Welcome back to IGCSE Science Academy. Hello everyone, today we are diving into a fundamental concept in biology unit 3, movement into and out of cells. This unit is crucial for understanding how life functions at a microscopic level. We are following the IGCSE Cambridge Biology 0610 and 0664, Chapter 3, Part 1. Every living cell is a busy factory, and for it to survive, it needs to constantly exchange material with its environment. In this session, we will break down the mechanics of movement through these five key areas. 3.1.1 What is diffusion? 3.1.2 Kinetic energy. 3.1.3 Cell membrane. 3.1.4 Living organisms, and 3.1.5 Factors and investigations with ATP practices. ATP practices is for the paper 6, which is alternative to practicals. For today's lesson, the learning objectives are: by end of this topic, you will able to describe diffusion as a net movement of particles down a concentration gradient. State that the energy for diffusion comes from kinetic energy of random particle movement. State that some substances move into and out of the cells by diffusion through the cell membrane. Describe the importance of diffusion of gases and solutes in living organisms. Investigate factors that influence diffusion: surface area, temperature, concentration gradient, and distance. Moving into our first core concept, 3.1.1, what is diffusion? By definition, diffusion is the net movement of particles from a region of higher concentration to region of lower concentration, that is down a concentration gradient, as a result of their random movement. The diffusion occurs naturally and does not require energy. For example, if you spray perfume in one corner of a room, after some time the smell separates throughout the room. This is because the perfume particles move from the area where they are more concentrated, means near the spray, to area where they are less concentrated. So another simple example, that is in plants, carbon dioxide diffuses from the air, which is higher concentration, into the leaves, which is low concentration, during the process of photosynthesis. Now, why does this happens? It's not because the particles want to move, it's a result of their random movement. If you look at the diagram, on the left we have a high concentration. Lots of blue particles crowded together. On the right, the green box shows a low concentration. Because these particles are constantly bouncing around, they naturally separate out into the space where there are fewer of them. Notice the orange arrow in the middle, net movement down gradient. It signifies the net movement, while individual particles might move in any direction. The overall flow is toward the low side. This movement does not go on forever. Through it continues only until equilibrium. Equilibrium is reached. That's the point where the concentration is equal on both sides and the particles are evenly distributed. Remember that down a concentration gradient, it's a favorite for examiners and the key to understanding how substances like oxygen and carbon dioxide enter and leaves our cells. Examiners give marks for examples. Choose simple ones like perfume separating in a room, ink separating in water. By these kind of examples, you get full marks for questions like this, that what is diffusion, define diffusion, explain diffusion. Now let's explain some key terms which are very important for the exams. First is concentration, the amount of a substance dissolved in a given volume. High concentration is equal to many particles, low concentration is equal to few particles. Then equilibrium. When concentration are equal on both sides, particles still move, but where is no net movement, equal numbers move each direction. Concentration gradient. The difference in concentration between two regions, the bigger the difference, the steeper the gradient. Diffusion moves down this gradient. Random movement. Particles have no preferred direction. They move in all directions constantly due to their kinetic energy colliding and bouncing off each other. Then net moment. The overall direction of movement particles move randomly in all directions, but more move from high to low than flow from low to high, creating a net moment. So these are the some key terms explained for your examinations. Remember, these are very important to memorize to get more marks in questions like diffusion, questions like concentration gradient. 3.1.2, that is kinetic energy and diffusion. Let's look at the engine behind this process. A vital key statement to remember is that the energy for diffusion comes from the kinetic energy of the random movement of molecules and ions. The cell isn't forcing this to happen, the particles are doing the work themselves. So remember this key statement. The energy for diffusion comes from kinetic energy of the random movement of molecules and ions. So what exactly is kinetic energy? The energy an object has due to its motion. All particles, atoms, molecules, ions are constantly moving and possesses kinetic energy. In physics term, we express this as kinetic energy is equal to directly proportional to its mass and directly proportional to square of its velocity, where m is equal to mass and v is equal to velocity or speed. Now, how does kinetic energy cause diffusion? Particles vibrate, rotate, and move randomly. They collide with each other and bounce off. This constant motion separates particles from areas of high to low concentration. No extra energy input is needed. It's spontaneous. Effect of temperature on kinetic energy. This brings us to a major factor, that is temperature. At higher temperature, particles gain more kinetic energy, they move faster, they collide more often, and therefore the diffusion rate increases. Conversely, at a lower temperature, particles have less kinetic energy, they move slower, and the diffusion rate decreases. If you want to speed up diffusion, just turn up the heat. Remember, diffusion is a passive process. It requires no energy from the cell, no ATP needed. So the passive rule, just remember this bottom line, diffusion is a passive process. It requires no energy from the cell, meaning no ATP is needed. It's all down to the kinetic energy already present in the particles. 3.1.3, diffusion through the cell membrane. We will explore how the cell acts as a gatekeeper, deciding what gets in and what stays out. First, let's talk about the cell membrane. Think of it as a selectively permeable barrier. It's primarily made of phospholipids bilayer, which you can see here in purple. It consists of polar hydrophilic heads on the outside and non-polar hydrophobic fatty acid tails on the inside. Because of this unique structure, the membrane does not just let anything through, it's very picky. So what can pass through? What gets a VIP pass to diffuse freely? So that small non-polar molecules are the ones that have it easy. This includes essential gases like oxygen, carbon dioxide, as well as water, fatty acids, and glycerol. However, if a molecule is larger or electrically charged, it can't just squeeze through the lipids. It needs help from a chemical channel protein which acts like a dedicated tunnel or pore through the membrane. Now, which way do these molecules move? The rule for diffusion is simple: high to low. Molecules naturally move from an area of high concentration where they crowded to an area of low concentration where there is more space. In our diagram, you can see a high concentration of blue molecules outside the cell. The net movement is towards the inside of the cell. While some back diffusion happens, the overwhelming flow follows the concentration gradient. It is important to remember that the cell does not control the direction of simple diffusion. It does not use energy to make this happen. It's a passive process driven entirely by the concentration gradient. Whether it's moving in or out, the molecules are just looking for balance. Here are some terms. First, the cell membrane, a selectively permeable barrier. It allows some substances to pass through but not others. Made of a phospholipid bilayer with proteins. What can diffuse? Small non-polar molecules, oxygen, carbon dioxide, water, fatty acids, and glycerol can pass freely. Larger or charged molecules need protein carriers. Which direction? Always from high concentration outside, low concentration inside, or vice versa. The cell does not control directions, only concentration gradient matters. So in simple, first diffusion is the movement of particles from an area where they are in high concentration to an area where they are in low concentration. In cells, the cell membrane controls what enters and leaves the cell. Small particles like oxygen and carbon dioxide can move across the membrane by diffusion. They move naturally, no energy is needed. If there is more oxygen outside the cell than inside, oxygen will diffuse into the cell until the concentration becomes equal. Diffusion through the cell membrane is equal to movement of substance from high to low concentration without using energy. 3.1.4, that is importance of diffusion in living organisms. We have seen how diffusion works at a microscopic level. Now let's look at the big picture. Why is this process actually vital for life? In living organisms, diffusion is the primary way we exchange gases, absorb nutrients, and remove waste. So the first part is about respiration and gas exchange. First, let's look in the respiratory and circulatory system. In lungs, oxygen diffuses from air into the blood while carbon dioxide diffuses out. Our alveoli are perfectly designed for this. They are only 1 cell thick and have a massive surface area to make this exchange as fast as possible. Gas exchange in leaves, photosynthesis and transpiration. So during photosynthesis, carbon dioxide diffuses into leaf cells through tiny openings called stomata while oxygen diffuses out into air. The spongy layer of the leaf creates large air spaces to ensure every cell is within reach. So here is the statement: stomata open to allow carbon dioxide in for photosynthesis. Spongy mesophyll cells have large air spaces to maximize diffusion distance. Then nutrient absorption. This is about digestive system. In human, diffusion is how we actually get fuel from our food. In the small intestine, digested molecules like glucose and amino acids move from the guts into the bloodstream through the villi. So in the small intestine, ileum digested food diffuses through villi into the bloodstream, high concentration in gut, lemon, low in blood. Then excretion, that is waste and signaling. The waste product is urea. Finally, diffusion plays a role in keeping our internal environment clean and our signals moving. Excretion: waste product like urea diffuses out of cells into the blood and eventually into the renal filtrate to be removed as urine. This prevents toxic build-up in our cells. Metabolic waste products: urea carbon dioxide diffuses from cells into blood and then into urine or exhaled air. Maintain high intracellular waste levels. Nerve impulse transmission: even our thoughts rely on diffusion. During an action, potential sodium and potassium ions rush across the neuron membrane down their concentration gradients, allowing electrical signals to travel through our nervous system. At last, oxygen delivery. The circulatory system. Oxygen diffuse into blood and then tissues, carbon dioxide out from tissues then to blood. In respiratory tissues, oxygen concentration in cells is low, used up in respiration, so oxygen diffuses from blood into cells, carbon dioxide diffuses the opposite way. From our very first breath to the way our brain processes this information, diffusion is the silent engine driving the most essential functions of life. 3.1.5 Factors that affect the rate of diffusion. Diffusion does not always happen at the same speed. There are four main accelerators that can make the process faster or slower. Let's break down the factors that determine the rate of diffusion. First, that is surface area. More surface area, faster diffusion. More area available for particles to cross at one. For example, alveoli, villi, and root hair cells all increase surface area for efficient diffusion. Like having more checkout lanes at a supermarket, more things can pass through it once. The first factor is surface area. The rule here is simple: more surface area equals faster diffusion. Think of it like a supermarket. If you have 10 checkout lanes open instead of just one, more people can pass through at the same time. This is exactly why structures like alveoli in the lungs, villi in the small intestine, and root hair cells in plants are shaped the way they are. They maximize the space available for particles to cross. Second factor is temperature. Higher temperature, faster diffusion. Heat gives particles more kinetic energy, so they move faster and collide more frequently. Body temperature, that is 37 degrees centigrade, is optimized for efficient diffusion, like cars going faster on a motorway when speed limits increase. We have temperature. Higher temperature leads to faster diffusion. Heat gives particles more kinetic energy, meaning they move faster and collide more frequently. It's like car on motorway. When the speed limit increases, everyone reaches their destination sooner. In the human body, our temperature is optimized at 37 degrees centigrade to keep these processes efficient. Third is the concentration gradient. A steeper gradient results in faster diffusion. Greater differences in concentration, greater driving force for net movement. Blood flow constantly removes absorbed substances, maintaining the gradient, like water flowing faster when there is a steeper slope in a river. The gradient is just the difference in concentration between two areas. The greater that difference, the stronger the driving force for movement. Our bodies actually work to maintain this. For example, blood flow constantly carries away absorbed substances, so the gradient stays steep. It's exactly like water flowing much faster down a steep mountain slope than a flat stream. For this distance diffusion path, stronger distance, faster diffusion. Particles must travel a shorter path to reach their destination. Thin membrane, for example, alveolar walls is equal to 1 cell thick allow very rapid diffusion. Like crossing a narrow stream as a wide river, the narrow stream is much quicker. Finally, we look at distance or the diffusion path. A shorter distance means faster diffusion. Particle needs to travel the shortest path possible to be efficient. This is why biological membranes like the walls of our alveoli are often only 1 cell thick. It's the difference between crossing a narrow stream versus a wide river. The narrow path is always much quicker. So to recap, to get the fastest diffusion possible, we want a large surface area, high temperature, a steep concentration gradient, and a very short distance to travel. In this slide, we are looking at a classic paper 6 alternative to practical or paper 5 practical. The agar cube experiment. This investigation is designed to model how the size of an organism affects the rate of diffusion. In exam terms, we are investigating the relationship between surface area to volume ratio and the rate of diffusion. Here is the experiment method. When describing this method in an exam, precious is key. First is preparation. We start with alkaline agar jelly stain with an indicator like a phenolphthalein. We cut this into cubes of varying side length, for example 0.5 cm, 1.0 cm and 2 cm. Second is procedure. Place all cubes into dilute hydrochloric acid at the same time. It is vital to start the stopwatch the moment the cubes are submerged. Then the observation and data collection. Observe acid diffuses in and changes the indicator color. Record the time for acid to reach the center for each cube. We observe the acid diffusing into the agar which changes the indicator color. We record the time taken for the acid to reach the center of each cube. Then calculation. Calculate surface area volume ratio for each cube. Then plot a graph, surface area volume ratio, x-axis as time for color change, y-axis. Safety, wear safety glasses, handle acid carefully. Here are the identifying variables: independent change size of agar cube, surface area, dependent measure, time for color change, diffusion distance, controlled keep same type of agar, acid concentration, temperature, volume of acid. Finally, analyzing the result, expected results. Here size in centimeters 0.5 multiplied by 0.5 multiplied by 0.5, and then 1 multiplied by 1 multiplied by 1, 2 multiplied by 2 multiplied by 2. Here surface area volume ratio 12:1, here 6:1, here 3:1. Diffusion speed here very fast, here slow. In your exam you may be asked to plot this. Remember, surface area volume ratio goes on the x-axis and time goes on the y-axis. The resulting graph will show that as the ratio increases, the time taken for diffusion decreases. Here are some ATP practice questions. Assessment for IGCSE Cambridge Biology Diffusion. First question from Assessment Objective 1. They may ask define diffusion, which may carry 2 marks, include net movement higher to lower concentration, random movement concentration gradient. Question 2. State the source of energy for diffusion, which carries 1 mark. This is from assessment objective 1 and kinetic energy of random particles movement not ATP. Question 3 from assessment objective 2, a student places cube of agar stained with indicator into HCl. Cube A is equal to 0.5 centimeter size, side cube B is equal to 2 centimeter side. Predict which cube will slow complete color change. First explain your answer. It carries 3 marks. Cube A higher 5 uh, cube A higher surface area volume ratio, more surface area relative to volume, acid diffuses to center faster. Question 4, explain why the rate of diffusion increases when temperature is raised. It carries 2 marks and this is from assessment objective 2. Higher temperature, more kinetic energy, particles move faster, more frequent collisions, faster net movement. Question 5, describe and explain the importance of diffusion in the human lungs. This is from assessment objective 3. It carries 4 marks. The answer tips: oxygen diffuses and carbon dioxide out. Alveoli features thin walls, large surface area, good blood supply, maintain gradient relates to gas exchange. Question 6, explain why diffusion alone would not be sufficient for gas exchange in large animals. This is from assessment objective 3. It carries 3 marks. The answer tip: large organisms have small surface area volume ratio, diffusion distance too great, too slow, needs circulatory plus respiratory system. Extended ATP practice exam style questions. Question 1 and question 2. Question 1 is data response which carries 6 marks. This is from assessment objective 2 and assessment objective 3. Question 2, that is application question, it carries 5 marks. This is from assessment objective 2. Dear students, practice these questions. These are very important questions from this unit. Write the answers and you can mention your answers in comment box. Here is exam tip. Use key term correctly for explained questions. Always include the mechanism why for describe questions. State the pattern trend clearly. Check marks allocation and write that many distinct points. So these are the exam tips. Once you practice and you done these questions, let me know in the comment box. To wrap everything up, let's look at this diffusion revision map. This is your at a glance guide to the entire topic, connecting the definition, the requirement, and the real world applications. Starting at the top left, what is diffusion? It is the net movement of particles from a region of high concentration to a region of low concentration. In other words, particles move down the concentration gradient. Remember that word net, it's a crucial because it describes the overall direction of movement, energy and the cell membrane. Moving to the top right, let's talk about energy. Diffusion is a passive process. This means it requires no ATP energy from the cell. Instead of it relies entirely on the natural kinetic energy of particles themselves. At the bottom left, we see the role of cell membrane. It is selectively permeable, meaning it only lets certain substances through like oxygen, carbon dioxide and glucose, while blocking others. This is how the cell controls its internal environment. Factors and real-world examples. On the right side, we recap the factors that change the speed of this process: surface area, temperature, concentration gradient, and distance. We see these factors in action across various biological systems. Examples include lungs for gas exchange, the gut for nutrient absorption, leaves for photosynthesis, and the kidney for filtration. Before we finish, look at the star at the bottom. This is a classic exam trap. Equilibrium does not mean no movement. When particles are evenly separate out equilibrium, they are still moving in both directions at the same rate. There is just no net moment. Diffusion is passive, continuous, and essential for life. So that covers our summary of diffusion. Keep these four factors and the definition in mind, and you will be well prepared for any practical or theory questions that come your way. Very essential topic for your exam. And that's a wrap on our diffusion series. If you found these breakdowns helpful for your exam preparation, please hit that like button and subscribe to the channel. It really helps me out and ensure you don't miss any of our upcoming biology practical and theoretical guides. Study hard, stay curious, and I'll see you in the next video.